So this is chapter 5, the cell cycle. Um, so cell goes through a phase from G1 to S phase and then G2 to M mitosis. And we call this G1S, G2 interphase, and then mitosis and cytokinesis M phase. And in average, cell cycle is 16-hour long. About 15 hours is spent on interphase and only 1 hour is spent on mitosis. And we get the increase in the gene material at G1. We have the 2N, which is represent the amount of DNA. And then on G2, we get the 4N. So G1, we have the start point, restriction point, and then we go through DNA synthesis on the S phase. And then G2, we have double chromatin and then and mitosis. We actually, that's when they get all pulled apart, going from prophase, metaphase, anaphase, and telophase. So until the G1 restriction point, they are G signal dependent, but after the G1 restriction point, G is a G signal independent. But once it's go, it's go. So cycling is like the traffic lights that gives whether to go or not, proceed or not. So you can think of like a keys for cars example, what starts the car or what starts the process of cell cycle. And then CDK, which is a cyclin dependent kinase. So what they do is add phosphate group. They're like the engine. That's what driving them. And CDKI, which is a CDK inhibitor, which they're like the brake. And checkpoint is what they're really doing is they're like asking, do we have enough resources slash energy? Are we big enough? Do we have enough space? Like, are we big enough? And are the DNAs we have are like of good quality? So how they regulate it is by telomeres and cell-to-cell signaling, and we'll go into more deep dive. One thing to note is that cell cycle are unidirectional. So after each cell cycle, those like cyclin and CDK, they're like degraded, so they can go back. So on G1, we have cyclin D and CDK4/6, and after that, we have cyclin E and CDK2. Last phase, we have cyclin A, CDK2, and G2, cyclin BA, and CDK1. They cell cycles so only goes forward, and if something goes wrong, we go through apoptosis. And how they end determines the cell cycle to go for a cell cycle or to start or not is by the bistable switch mechanism, which is basically an on and off switch. They have the double negative feedback, so inhibitors keep inhibiting activators. Activators, as when they're active, they inhibit the inhibitor so they can stay on. So it's either on or off. So for checkpoints, at G1 restriction point, CDKs are serine threonine kinase. Protein concentration of CDK does not fluctuate during cell cycle. Cyclin CDKs can be inhibited by CDKIs. Phosphorylation and dephosphorylation of CDK at inhibitory or activating site regulate CDK activities. So G1 cyclin CDKs are what controls the entry into S phase. The commitment point, we call it in G1, we call it restriction point, and inhibit E to F. And S phase cyclin CDKs, they replicate DNA only once, and because they only want to go through it like once, right? So how they do it is they bookmark what they have done and throw away the reused one through the polyubiquinate. And CDKIs, which is cyclin CDK inhibitor, they arrest cell cycle progression and often tumor suppressors as well. they are often tumor suppressors. And they have P16 and P21 as a kind of CDKI. And G2, they look for DNA damage. And CHK2 or CDK2 is like the checkpoint for kinase. Um, so DNA damage causes ATM, and ATM goes to CHK2, and then it goes to P53, and P50 goes through the feedback loop with M2M2 and WIP1 and activate P21, and P21 inhibits a cyclin B and CDK1, which is in the G2 phase. In P53, the transcription factor depends on the state of cell. You can either degrade it if the cells are okay, or it can P53 can forces apoptosis. And we have the M checkpoint, mitosis checkpoint. It can stop if the proper attachment of the chromosomes are not there. And APC, which is an anaphase promoting complex, plays a huge role in cleaving chromosome with separase or degrading with cyclins to reset the cell cycle. If there's a too many cyclin P1, we can cause cancer. And for a drug, we can do like a CDK inhibitor. What they're doing is basically stopping the engine. So they're stopping the cell division, or CHK inhibitor, which is checkpoint kinase, and they can forcefully divide and cause apoptosis. Or microtubule inhibitors, they don't assemble or they're too stable to separate, so they make it cytostatic and do not cause cell division. And that's about it.